Hello, my name is Jeff Disson, and this is my microcosm PowerPoint presentation for General Ecology EVSP 416. Microcosm Overview. I started this project because I was curious to see if I could create a little self-sustaining system inside of an airtight glass jar. I hope to learn more about autotrophs and be able to see if they could create their own energy using photosynthesis. I also wanted to see if I could get any heterotrophs in there as well, but I'll show you later on that was pretty much unsuccessful. So microcosms can be used in ecology to study on a smaller scale what is happening in a larger area by taking small sample of the soil or water. I got the sand from a local man-made fishing pond in my community. I live in the middle of the desert and I am pretty busy, so trying to travel an hour or more to the lake or some small rivers that we have here was pretty difficult. So I went to a man-made lake, got some water there and some materials from the side, and I'll show you what I came up with. My hypothesis is that algae will grow and become self-sustainable within the airtight sealed jar, and I will see living organisms swimming around. And let's see how it turned out. My methods and materials. I built my microcosm by putting some sand in the jar first, then filling it with water. Where I got my material, there are a lot of ducks and geese, so I also grabbed what looked like duck poop or geese poop covered in algae. It was the only green algae that I could find in the area. I'm not exactly sure what it was because I didn't actually, tear it apart and look at it, but I used about 5 centimeters of sand gathered from the bottom. I scooped that up, and then about 550 milliliters of water. The microcosm project ran from September 9th, 2021 to October 24th, 2021. The microcosm received direct sunlight for about 5 hours of the day and indirect sunlight for about 7 hours of the day. The temperature inside where it was located was 22 to 25 degrees Celsius. It didn't vary that much because I keep the AC on and keep it fairly comfortable in the house. I didn't put it outside because even though it is September going into October, Phoenix is still really hot in the triple digits still, so I didn't want the water to get too hot and just kill off everything. My observations and data. I observed many changes in my microcosm. It started off with clear and clean sand and clear and clean water, but as the time went on, the algae sunk to the bottom and floated to the top a few different times because of air bubbles. I had a lot of questions in the chat from a few different people, like talking about the air bubbles, and so I looked it up, and it's actually just because of the photosynthesis during the day where it produces oxygen and then it just helps it float to the top, and then at nighttime it can sink down and then repeat the process. But mine floated to the top, stayed there for a while, and then eventually just sunk down to the bottom and never came back up. As time went on, the algae sunk to the bottom, floated to the top a few different times. This is because of air bubbles the algae produced through photosynthesis and caused the algae to float. At night, when the algae was not producing anymore, it would sink to the bottom. Oxygen, it would sink down to the bottom until finally it stopped floating altogether and came to rest at the bottom. The top picture is the first week I had my microcosm, and the bottom picture is one of the last photos I took almost 7 weeks later. As you can see in the pictures, the algae has fully taken over the jar, penetrated the sand, and the water started off nice and clear, but later became murky and dirty looking. I'd like to point out that there is some weird like spider webs growing attached to the side of the jar, and the algae went into the sand and just pretty much took over the entire microcosm. The top picture there looks pretty clear and clean, and then the bottom picture is a little nasty looking. I did see some swimming organisms for about a day, little tiny specks. I couldn't tell what they were, but I could see them moving around, and then the next day I tried to find them and they were gone. Also, I saw like a couple worm-like creatures on the side of the glass. There was about 2 of them and they were moving, and then the next day they were gone as well. So I don't think they lasted very long, but there definitely was some living organisms in my microcosm. Not sure if it got too hot from the direct sunlight or just the algae took over. I don't really know for sure what happened to them, why they died off. Major findings. I found the algae did overtake the entire system, microcosm. I am not sure what type of algae, as I stated before. It did produce a large amount of oxygen, and it continued to produce a large amount of oxygen throughout the entire process. My observations did support my hypothesis to a point. I did see heterotrophs, but only a couple and not for very long. The algae did become self-sustainable and survived on its own for the life of the microcosm, and it seems to be doing really well, and it continues to produce oxygen. I did not get any. It was airtight. I did not get any water dropping or anything like that, so it seems like a nice little spot for the algae to grow. Microcosms in context. So concept number 1 that I learned was adaptation to the environment. The algae adapted very well and overcame its environment and thrived in the microcosm. Day after day, it would use photosynthesis to produce oxygen and end up taking over the entire microcosm. Concept number 2, what we can learn from microcosms. Because ecology is a hard field to study and come up with definitive answers, we can use microcosms to study on a smaller scale in a controlled environment to get a better understanding of what happens in the ecosystem. Concept number 3 is biodiversity. Within this microcosm, it started off diverse with autotrophs and heterotrophs, but as time went on, the autotrophs took over the microcosm and left the living conditions for the heterotrophs uninhabitable. Much like human population increasing, putting large amounts of pressure on the flora and fauna because we overconsume and overbuild and take over everything. So biodiversity is kind of failing there. So for further research, I would have liked to actually go to a river or a lake instead of a man-made pond area. I'd like to go to like a forest or something and actually get some good materials to put in there. Maybe find some heterotrophs to put in there and see if I can actually sustain a little microcosm with living creatures and see if I could get it to be like its own little ecosystem that would thrive. I would really like that. Next time I would get a larger jar for my microcosm and add some foliage from the riverside and maybe some sort of snail or mussel to see if they can survive and even produce. I would set it up during the fall instead of the summer because it gets so hot here in Phoenix. I think that may be one reason why my microcosm didn't support the heterotrophs, is just because it was really, really hot. I'm pretty sure the direct sunlight on the jar heated up the water, and it might have been too hot. I'm not exactly sure, but that's my hypothesis for that. Microcosm in context. Microcosms are generally relevant or extremely relevant for use in general ecology. They can be used to study biodiversity. We can use them to study producers and consumers. We can use them to study disturbances in ecosystems, and they can be used to study the roles of key species. Microcosms are used a lot in a lot of different sciences to study a lot of different things, and we can take our microcosms and build them how we want to see what different outcomes we can get with different varieties of autotrophs, heterotrophs, and whatnot. And these are my references, and that is my microcosm in a nutshell, or should I say a glass jar.